Good morning, everyone. In the last session, we were discussing about the biodegradation, the definition of biodegradation, and what constitute the biodegradable matter. And we were also discussing about the bioremediation of pollutants utilizing the biodegradation abilities of microorganisms. In today's session, we will continue our discussion on the microbial degradation, and in the next half, we will discuss about the biopesticides. Bacteria with the ability to degrade hydrocarbons, they are called as hydrocarbon degrading bacteria. And the biodegradation of hydrocarbons, it can occur under aerobic and anaerobic conditions. So the strains which are usually used are gram-negative bacteria, and most of them belong to the genus Pseudomonas. So the biodegradative pathways have also been reported in bacteria from the genera such as the Mycobacterium, the Corynebacterium, the Aeromonas, Rhodococcus, and Bacillus. Although many bacteria they are having the capability or they are able to metabolize the organic pollutants, a single bacterium does not possess the enzymatic capability to degrade all of the organic pollutant or even most of the organic pollutants that is present in the target site. So mixed microbial communities have the most powerful biodegradative potential, right? So a single bacterium does not possess the enzymatic capability to degrade all the organic pollutants or even most of the organic pollutants when it is defined in a particular or specific polluted soil or target site. So mixed microbial communities have the most powerful biodegradative potential for the reason that the genetic information of more than one organism is necessary to degrade the complex mixture of organic pollutants present in the contaminated sites. So, right. So the genetic information from a mixed microbial communities, that information which is present in the mixed microbial community is necessary to degrade the complex mixtures of the organic pollutants. So that means the organic pollutants present in the target site or the contaminated site. And both anaerobic and aerobic bacteria are capable of biotransforming the PCBs. Right. So higher chlorinated PCBs are subjected to reductive dehalogenation by anaerobic microorganisms, and lower chlorinated biphenyls, they are oxidized by aerobic bacteria. So we have two categories over here. The higher chlorinated PCBs, they are subjected to reductive dehalogenation by anaerobic microorganisms, and lower chlorinated biphenyls, they are oxidized by aerobic bacteria. Right. So heavy metals. What about heavy metals? Heavy metals cannot be destroyed biologically. That means no degradation or change in the nuclear structure of the element occur, but they are only transformed from one oxidation state or organic complex into another. Is that clear? So besides bacteria are also efficient, these bacteria they are more efficient in heavy metal bioremediation. So microorganisms they have developed the capabilities to protect themselves from heavy metal toxicity. So it is an innate characteristic of the microorganisms which they have developed, or they have the capability to protect themselves from heavy metal toxicity around their living habitat by various mechanisms such as adsorption, it could be uptake, it could be methylation, oxidation, and reduction. So heavy metals cannot be destroyed biologically, thus no degradation or change in the nuclear structure of the element occur, and they are only transformed from one oxidation state or organic complex into another. And microorganisms, they have developed the capabilities to protect themselves against the heavy metal toxicity. The mechanisms which are involved is, that means how do they protect themselves against heavy metal toxicity? The mechanisms involve adsorption, uptake, methylation, oxidation, and reduction. Right? Now speaking about the gen, that is genetically engineered microorganisms. Now the molecular biology has offered tools to optimize the biodegradative capacities of microorganisms. So this has accelerated the evolution of new activities and construct totally new pathways through the assemblage of catabolic segments from different microorganisms. Right? So it is because of the tools, or with the advent of the tools in the area of molecular biology, the tools to optimize the biodegradative capacities of microorganisms has paved way for genetically engineered microorganisms to be used in biodegradation process. And it has accelerated the evolution of new activities and to construct totally new pathways through the assemblage of catabolic segments from different microorganisms. Right. Now the genes which are responsible for degradation of environmental pollutants or organic pollutants, they have been identified. Right. So the pollutants, environmental pollutants, say for example toluene, chlorobenzene, acids, and other halogenated pesticides and toxic wastes. So all these constitute the environmental pollutants. And for every compound, one separate plasmid is required. Is that clear? So the genes responsible for the degradation of the environmental pollutants, say for example toluene, chlorobenzene, and other halogenated pesticides and toxic waste, they have been identified. And the interesting feature is for every compound one separate plasmid is required. It is not like that one plasmid can degrade all the toxic compounds of different groups. So separate plasmid is required for every compound. So now the plasmids, they are grouped into four categories. The OCT plasmid, number one. OCT plasmid is the one which degrades octane, hexane, and decane. Octane, hexane, and decane. And now the second one is XYL plasmid. So this plasmid degrades xylene and toluenes. And number three, CAM plasmid, that decompose camphor. And number four, NaH plasmid, which degrades naphthalene. Is that clear? So for every compound, that is, it could be the toluene, chlorobenzene, or other halogenated pesticides and toxic waste which have been identified, one separate plasmid is required. So plasmids are grouped into four categories. This is in a broader sense. Number one, OCT plasmid, which degrades octane, hexane, and decane. XYL plasmid, which degrades xylene and toluenes. CAM plasmid, that decompose camphor, and NaH plasmid, which degrades naphthalene. Is that clear? Fine. Now, what are the obstacles that are associated with the use of genetically engineered microorganisms in the bioremediation applications? While genetic engineering has produced numerous strains which have the capability or which are able to degrade otherwise intractable pollutants in a petri dish or in a bioreactor, the practical translation of this research into actual in situ bioremediation practices is very little or quite scanty. Less information is known about this process, right? So the major issue in this respect is the growing realization that the strains and bacterial species that most frequently appear in traditional enrichment procedures, they are not the ones performing the bulk of biodegradation in natural niches, right? So the major issue is that the most frequently appearing microorganisms in traditional enrichment procedures, they are not the ones that are performing the bulk of biodegradation in natural niches and may not even be any good as bioremediation mediators, right? So in addition, the developments or the recent advances in the area of the recombinant DNA technologies, it has paved the way for conceptualizing something called as suicidal genetically engineered microorganisms. That is, yes, gens, right? So it is something very recent concept in the area of the rDNA technology, the conceptualization of suicidal genetically engineered microorganisms, that is SGEMs, to minimize such anticipated hazards and to achieve efficient and safer decontamination of polluted sites through these suicidal genetically engineered microorganisms. So this is something very interesting, and it is also innovative to understand how the genetically engineered microorganisms, especially the S-gems, play a role in the process of biodegradation. In some cases, whether the introduced bacterium is recombinant or not making any little or any difference or a little difference. So, because the problem is that of the implantation of the foreign microbes in an unfamiliar territory. So, it is not just like you go and plant the microorganisms in the contaminated site and you expect that biodegradation to happen. So, the obstacle is that the bacterium, whether it is recombinant or from the natural sources, whether it makes some difference or may not make any difference, because it is the problem of implantation of foreign microbes in an unfamiliar territory. So as soon as you implant in an unfamiliar territory, the microorganisms may grow or it may not grow, and even if it grows, it may not be able to make any big difference in terms of the bioremediation. So this is another important obstacle associated with the use of genetically engineered microorganisms. So the introduction of bacterial biomass in an existing niche may create a palatable niche for protozoans that prevents the bacterial population to grow beyond a certain level. So in the new niches, there could be some other factors which actually prevents the bacterial population growth beyond certain limits, say for example the protozoans. So this is an important obstacle. So bacterial biomass cannot be just increased in exponential manner. So there could be some a palatable niche, say for example the protozoans, that prevents the bacterial population growth to a certain level. So ingenious approaches have been developed to circumvent this problem. So we have to overcome this problem. So the bacteria should be capable enough to grow as soon as it is implanted in an unfamiliar territory, and it should overcome the palatable niche exerted by the protozoans as an example. So ingenious approaches have been developed to circumvent this problem, including say for example encapsulation of the inoculum in a polymeric matrix or protection in plastic tubing. So this is one important ingenious approaches to overcome this kind of obstacles, right? And now the next major problem encountered is the successful bioremediation technology, which pertains to hostile field conditions for the engineered microorganisms. So the main concern is to construct the genetically engineered bacteria for field release in bioremediation with an adequate degree of environmental certainty. So the bacteria or the bacterial biomass should be capable enough to acclimatize to the field conditions. So efforts should be made to examine the performance of engineered bacteria in terms of their number one, survival, number two, potential of horizontal gene transfer, so which may affect the indigenous microflora within the complex in an unknown territory or the environmental situations. So this is another important problem, a major problem encountered when we look for a successful bioremediation technology. That means it is about the acclimatization of the microorganisms to the field conditions. So we should be having this concern for field release of the bacterial population with an adequate degree of environmental certainty. So environmental acclimatization of the biomass or the bacterial biomass that is released should meet and should get or should involve completely in the process of bioremediation, and efforts to examine whether the survival rate is good, whether any potential horizontal gene transfer is happening, which may affect the indigenous microflora of that particular niche, so or within the complex environmental situation. So all these factors need to be taken into consideration, or it has to be overcome, right? And next we move on to a new concept of unit 2, that is biopesticides, especially with respect to the pest control and the utilization in pest management formulations application technology or the method. So the biopesticides, as we all know, are naturally occurring substances from living organisms, that is natural enemies, what we term, or their products. It could be microbial products or it could be phytochemicals, or their byproducts. So we refer this to as semiochemicals that can control pest by non-toxic mechanisms, right? So they are defined as naturally occurring substances from living organisms, that is natural enemies, to protect themselves from the natural enemies within that habitat. So these are naturally occurring substances secreted from the living organisms or their byproducts, that is phytochemicals or microbial products, or their the semiochemicals, the byproducts that control pest by non-toxic mechanisms. So, Organization for ECD, that is Economic Cooperation and Development, which is where the headquarters is in Paris, the France, which was founded in 1961. This organization viewed biopesticides as manufactured mass produced agents derived from natural sources, living microorganisms, and sold for the use to control pest, right? So how do the ECD, that is Economic Cooperation Development Organization, define biopesticide? They view the biopesticide as manufactured mass produced agents derived from natural sources, living microorganisms, and sold for use to control the pest. Okay. Now the concept of biopesticides is that they are obtained from living organisms, their products or their byproducts, which we refer to as semiochemicals. Now if you just look at the categories of the biopesticides, they fall under four major categories. Number one, microbial pesticides. Number two, plant incorporated protectants, abbreviated as PIPs. Number three, biochemical pesticides. Number 4, semiochemicals. Is that clear? So if you just categorize the biopesticides, they are or they fall into four major categories. Number one, microbial pesticides. Number two, plant incorporated protectants, that is PIPs. Number three, biochemical pesticides. Number four, semiochemicals. So what are microbial pesticides? We'll just try to understand each of these categories of biopesticides. So these microbial pesticides, these consist of microorganisms such as the bacterium, virus, fungus, protozoans as active ingredients, so which are used for the biological control of plant pathogens, pestiferous insects and weeds. Right. So these microbial pesticides, they consist of microorganisms such as bacterium, virus, fungus, protozoans as active ingredients. So the most widely used microorganisms in the development of biopesticide is as we all know, is the insect pathogenic bacterium, that is Bacillus thuringiensis, or abbreviated as BT. You might have heard about BT cotton, BT brinjal. So that is about Bacillus thuringiensis. So that comes under microbial pesticides. So they consist of microorganisms such as bacterium, virus, fungus, protozoans as active ingredients to be used for the biological control of plant pathogens, pestiferous insects and weeds. So this Bacillus thuringiensis, which is an insect pathogenic bacterium, it serves as an insecticide for most of the Lepidoptera, Coleoptera and Antidaptera. So it is in a way it covers most of the genus of the insects, right? And next we have the biochemical pesticides. These biochemical pesticides, they are known as herbal pesticides and are naturally occurring substances used for controlling the pest through a non-toxic mechanism, right? So they are known as herbal pesticides and they are naturally occurring substances used for controlling the pest through non-toxic mechanism. And next we have the plant incorporated protectants, that is PIPs. Now what are PIPs? PIPs, they are also known as genetically modified crops, and they are the biopesticidal substances produced by plants from genetic material that have been added or incorporated into their genetic makeup. So we should be very careful in understanding the differences between different categories of the biopesticides. So plant incorporated protectants, which are also known as genetically modified crops, and they are biopesticidal substances which are produced by plants from genetic material that have been added or incorporated into their genetic makeup. And the fourth category is the semiochemical. So a semiochemical by definition is a chemical signal produced by one organism and which is usually insects, which cause a behavioral change in an individual of the same or different species. So for example, in crop protection, the most widely used semiochemical are the insect pheromones, which serve as a signal to communicate with others in their species for a number of reasons and synthesized for pest control by mating disruption. So there are four categories, as I said, and we'll just try to refresh all these category of biopesticides once again. So the four categories are microbial pesticides and plant incorporated protectants, biochemical pesticides and semiochemicals. So the microbial pesticides, so be careful in understanding the difference between these four categories. These microbial pesticides, they contain or consist of microorganisms such as bacterium, virus, fungus, protozoans as active ingredients, And then we have biochemical pesticides, which are the herbal pesticides. That is, they are naturally occurring substances and they control pests through a non-toxic mechanism, very very important. And then we have the PIPs, which are also known as genetically modified crops, or and they are the biopesticide substance produced by the plants from genetic material that have been added or incorporated into their genetic makeup. And then we have the semiochemical, and by definition it is a chemical signal produced by one organism, usually the insects, which causes a behavioral change in an individual of the same species or of the different species. So, with this, we have tried to understand the biodegradation process and the role of genetically engineered microorganisms in the biodegradation process. And we have also discussed about the biopesticides, what are the biopesticides, and what constitute a biopesticide, and what is the role of microorganisms in biopesticide, and how the biopesticides are categorized, what are the different categories of biopesticides. So we will continue our discussion on biopesticides in the next session. Till then, thank you very much for your listening.